DNA profile is a mechanism for finding a suspect against a list of different criminals or common people. For that thing, we have a CODIS database, which is basically used by FBI in USA to identify different criminals which are suspect in some different crimes. So what is this CODIS and what are its different parts? Let's see this thing. Number one, CODIS stands for Combined DNA Index System. So Combined DNA Index System means that we have a list of DNAs extracted from different people, and then those DNA information is stored in a database format. So basically it is a DB of DNA profiles for both the criminals और voluntarily samples. ऐसे लोग जिन्होंने अपनी मर्जी से अपने DNA samples जमा करवाए हैं for experimental और investigation purposes. So idea is DNA is collected from a crime scene. Then this DNA profile is run, and if that DNA is matched against some suspect, then criminal is caught. So CODIS basically, it is similar to our fingerprint system, which is being used in our countries, of course, and across the world, where we have a thumb system. We take, whenever we want to record something, we just take the impression of the thumb, and then if there is some crime or some investigation, that thumb impression is checked against that record and the culprit is found out. But CODIS is based on comparison of DNA instead of fingerprints. Fingerprints are basically based on our phenotype information. Phenotype means our physical information. On the other hand, DNA information, it is based on genotype. Genotype means genes information. So what are different indexes or information available in CODIS? So number one information is of offender index. Offender means those people who have convicted some crime. Similarly, arrestee index. Arrestee index means people who have been arrested because of doing some crime. Then we have forensic index. So forensic index means that index which is generated by taking DNA samples of the different culprits and arrestee people. Then missing person index. So missing person index, because those people are missing, so their DNA cannot be gathered directly from those people. So for that purpose, relatives of those people are usually taken, such as their parents index, their parents DNA, and then this information is used to finding those missing persons. And if that before missing, if the DNA is available, then their DNA is recorded in the CODIS database. So what are the benefits of STR in CODIS? What is STR? STR is short tandem repeat, which is basically a repetition of a similar sequence in a DNA sequence that is basically useful for finding a person in CODIS database. So what happens is we have forensic sciences. Forensic sciences are those people which are involved in collecting, storing and retrieval of this information from CODIS database. What they do, they take advantage of the population's variability in STR length. STR length means short tandem repeat length would be different for different people. Then based on this length, people or biologists or forensic experts are able to identify or differentiate one person from the other person. For example, two individuals with same 13 loci DNA profile, there would be only one person in 1 billion which have the same information as shown by this example. There are two individuals with same 13 loci in a DNA nucleotide sequence. So number of STR in CODIS, basically locus or on chromosome 7. So 13 code STRs vary in length from 100 to 300 bases. So for instance, this D7S820, which is a short tandem repeat, it is very simple and it contains roughly 10 to 11 repeats of short tandem repeat STRs. So how it has inherited or from where it has taken this information, we can say that it would have inherited a copy of this tandem repeat with 10 different GATA repeats, and these repeats can be from his parents, from his father, or the 11th could be from his mother. So here I have given a complete working of CODIS, how it works. So if I divide this thing, so for CODIS basically, since it starts from the extraction of a DNA, so for this thing it needs an information which should be in the form of some cell. So this cell can be a bone cell, blood, hair, body, or fluid, which is shown in this box. So once we have this information of a suspect, then we take this to a sample collection lab. From this lab, we apply different mechanisms which are for extraction of a DNA from a given sample. So these are our samples and this is our DNA extraction module. So once this DNA is extracted, it is taken to this DNA profile. So this DNA profile basically, it is an annotation profile. So in annotation profile, what we do, we usually put names of suspects with that specific DNA and its different properties. So once this DNA profile is generated, then we have different tools which we have already studied in this course and also in different courses, which are such as for multiple sequence alignment, BLAST, and different variants of BLAST, cluster W, and so on, which are written in this box. So these tools are used for sequence alignments, and then once these sequences are aligned, if there is some match, then we say that okay, so what are other information which can be triggered or extracted from this part. If there is no match, then we say okay, he is not a culprit, he should be removed. And if it is culprit, then we want to try out different other information which can be extracted by this DNA, such as gender determination and other information such as relatives, parent-child relationship, and so on, which is shown in this part.